how your DNA leads to protein synthesis. Proteins are involved in a lot of activities occurring inside your body, such as transport, structure, protecting the body, enzymes, and so much more. Instructions to make protein are contained in our DNA. All of your cells have DNA, which resides inside the nucleus. DNA has genes, which are portions of DNA that code for a protein, and when a gene is expressed, a specific protein will be produced through protein synthesis. In protein synthesis, there are two steps, the first being transcription and the second being translation. Transcription is the process of enzymes using one strand of DNA within the gene as a DNA template strand. There are three steps to this process: initiation, elongation, and termination. During initiation, the promoter region of the gene acts as a function site where an enzyme called RNA polymerase can bind. The DNA strand unzips using the help of a protein called helicase. The stranding reading from 5' to 3' is used as a DNA template strand. During elongation, the RNA polymerase slide along the 5' to 3' strand, pairing up complementary bases and linking nucleotide to the 3' end of the growing RNA molecule. mRNA is completed when the RNA polymerase reaches the termination stage, and the mRNA and RNA polymerase detach from each other. The mRNA includes regions that are called exons and code for protein, and non-coding regions called introns. In order for the mRNA to proceed to transcription, the mRNA must remove the introns using the help of proteins. Spliceosome mRNA leaves the nucleus through the nuclear pore, where it can enter the translation stage. Translation. How does the strand translate into a protein? It begins with initiation. The process involves several enzymes and several types of RNA molecules. The nitrogenous bases on the mRNA are grouped into three bases called codon. The genetic code includes 64 codons, which codes for specific amino acids. There are specific codons that code for start, AUG, and code for stop, AGU, UAG, and UAA. Translation occurs when the small ribosomal subunits binds to the start portion of the mRNA. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Refer to the codon chart. 3 of the 64 codons do not code for an amino acid. Instead, it codes for the chain to stop. tRNA picks up an amino acid, and the mRNA codon pairs up with the anticodon of the tRNA, forming a complementary base pair between the two. Only 2 tRNA amino acid complexes can arrive at an amino acid at a time. After the tRNA binds with the start codon, the large ribosomal subunit then binds to form the translation complex, completing the initiation stage. Elongation occurs as the process continues. Another tRNA will enter the ribosomal subunit from the right, forming complementary base pairing with its corresponding sequence, forming a peptide bond is formed between the amino acid. Elongation proceeds until a stop codon is encountered, which then a release factor binds to the stop codon, and the polypeptide then detaches from the tRNA, therefore concluding the protein synthesis process, in which being the termination stage.